Hello everyone. Have you heard about ICRPG entrance exam, which provides the fellowship of 12,650 per month plus contingency of 6,000 per year, earlier known as JRF, for the top rankers in this ICRPG entrance exam? So every year 24,000 plus students write this exam and only top 10% of the students will get this prestigious fellowship. So whether you are pursuing your BSc Agriculture from the public university or from the private universities, you can still pursue your masters from the India's premier institutes like Indian Agriculture Research Institute if you are securing the top rank in this exam. This becomes super easy joining AgriAddict, a platform for all agricultural exams. So we hold a proven track record of highest number of JRFs in the recent years, where 7 out of 10 students from the stream of Plant Sciences were our students. So we are a team of PhD and the MSc scholars from Indian Agriculture Research Institute and other top agriculture universities with the top rank in this ICRPG and PhD entrance exams. So we offer the complete online master course for the 4 major subjects in agriculture, starting with agronomy, entomology, plant sciences and physical sciences, and also the complete mock test bundle along with the doubt clearing sessions for the 6 different subjects in agriculture, starting with agronomy, entomology, physical science, plant sciences, social science and horticulture. So every year we provide the live interactive classes along with the recordings, our own study materials which are compiled to make it best and precise, regular mock test series according to the standards of ICAR, previous year discussion papers along with the PDFs with solutions, short and crisp notes for the key and memory based content, one-to-one doubt clearing sessions, and also we provide the complete revision series during the peak time of the exams. Already 2000 plus students are benefiting from our course, and you can see hundreds of positive feedback and appreciation for our content in the Play Store and the website. So if you have not yet registered to our online courses, visit our website www.agriaddict.in or download our Android mobile application AgriAddict and enroll today. So the validity of the course will be up to the day of the exams. So if you want to extend the validity of the course and for any other queries, you can contact us to the below displayed WhatsApp number. Keep learning. Thank you.
Hello everyone, I'm Arifasad from Team AgriAddict. So India has finally announced the dates for ICRPG and PhD entrance exams 2023. So it is unexpected that this year exams are very near. So in this video, I'm going to explain in detail about how you can cover the plant science in 15 days, what are the important topics to be covered, and how you can make yourself productive. So stick until the end of this video and take a full benefit out of it. Let's get started.
Okay, I will be starting with the genetics and covering all other subjects sequentially. Okay, so before I start, let me assume that there are 3 categories of students right now who are preparing for ICAR JRF exam. So first category will be who are very up to date and who are now revising the content, and second category who have not finished the full syllabus or they have just covered the 50 or 50% of the syllabus right now, and last category they are still in the initial phase. So in this video, I'm going to address all these categories of students and tell you what exactly you can do in these 15 days so that most of the content is covered. So to begin with genetics, so for genetics, as you all know, the standard book is BD Sync. So whatever you cover, you should cover from BD Sync only. So only for the revision, you can use your class notes or Agri materials or any other source from any other source that you get. Okay, but for the clear cut understanding, you should go with the BD Sync. So for now, even in the 15 days, you can cover this BD Sync only if you target the important topics that I am going to discuss today. So I will just go chapter by chapter and tell you like what are the different concepts that you have to give the importance or the priority. So starting with the history, from the history part, just concentrate on the scientists and their contributions and the important years. So this is the first thing. Secondly, concentrate on the evolution theories that are mentioned. So if you are covering these 2 parts, that would be enough. So second, cell ultrastructure. So what you can cover from this chapter? So when I talk about the cell ultrastructure, you need to have a clear cut understanding about the differences between plant cell and the animal cell. So this is the first thing. And next, the important functions of different cell organelles. And if there are any enzymes that are involved or associated with these cell organelles, just go through it once. So you just need to concentrate on the different cell organelles and their important functions. So that would be enough for cell ultrastructure along with the plant cell and animal cell difference. So this is present in a table, just go through the table once. Third chapter or third unit is chromosomes. Talking about the chromosomes, know about the morphology, that is size, shape and different types based on the centromere you get. and also we have different models of the chromosome about the organization, like nucleosome solanine model, and we have folded fiber model, and the scientist associated with them, like who gave these models. So these are important, just go through them once. And also the organization of the chromosomes, how it is organized, or how the chromatin is folded into chromosomes. I have a clear cut idea about these things in the chromosome chapter. Next, it's about cell division. So cell division basically means mitosis and meiosis. So make sure to read the table where exactly the difference between these two process is given. So make sure to read it very carefully and revise it from time to time. And talking about the mitosis, know about the different stages both in meiosis and mitosis, and what actually happens in different stages. What are the different events that are taking place in these stages? Like example, the crossing over, synaptic complex, and about the ploidy level. What is the ploidy level in case of mitosis, in the meiosis, and the number of daughter cells that are formed. So these things you need to concentrate. Okay, and also keep a track on the chromosome structure of the chromosomes, that is changing from chromatin to chromosome back to chromatin. At what stage this chromatin unfolds or fold itself to become chromosomes, and at what stage it again becomes the chromatin in case of mitosis and the meiosis. So that would be more than enough from this chapter. So next we have chromosomal aberrations. So mainly chromosomal aberrations of two types, that is numerical and structural. So know about their origin, how these aberrations are actually occurring. So this is the first point. Secondly, know about how they behave during the cell division, how they are dividing, how these chromosomes divides during the cell division if they are aberrated. And thirdly, about the genetic effect, what type of effect it is showing on organisms. So if you are concentrating on these three different things, that would be sufficient. So this is all about the first 5 chapters. Next we'll start with Mendelism. So what are the different things that are very important from Mendelism? Starting with laws of Mendel, know about 2 laws, and there are some exceptions to these 2 different laws, know about them. And about dihybrid and monohybrid cross, F1 and F2. So outcome of the F2, like total number of populations, we have different formulas for genotype, phenotype. Just make sure to know about all these things. So that would be more than enough from these laws of Mendel. And also you might expect like questions from, or problem based questions from this particular chapter based on monohybrid and dihybrid cross, to make sure to understand about these crosses. Okay, so next in the Mendelism, secondly we are concentrating on gene interaction. So you can actually expect at least one question from this gene interaction. So we have different types of gene interactions. So what you should concentrate is test cross ratio of them, and also F2 ratio. So if you are not going in detail about all this interaction, at least try to remember the test cross ratio and the F2 ratio. If you are remembering the examples along with the actual mechanisms why these ratios are coming, that would be beneficial for you. If not, just at least try to remember the test cross and F2 ratios. And also in this we have probability chapter. So no need to read this probability chapter in detail. Just know the applications of how this probability can be applied to know the outcome of Mendelian crosses or Mendelian segregating populations. So to know these things, like just know the application of the probability in this particular case, where you can expect the problem based questions. They will just give some crosses, and they will just ask you to predict the outcome of the cross, or directly they will ask you the probability. Next we have an important chapter which is linkage and crossing over. So in this linkage and crossing over, concentrate on 2-point test cross and 3-point test cross. The formulas to find single crossover, double crossover, and also the concept of coefficient of coincidence, and also the interference, what they actually mean and what is the formula to find out. So if you are covering these things, So that would be enough. This is the first thing to cover in the linkage and crossing over. And secondly, just concentrate on the theories or hypothesis on crossing overs, like breakage and reunion theory we have, and also we have copy choice theory, and also scientists who gave them. So just make a note of it. So that would be enough from linkage and crossing over. And another important chapter is multiple alleles. When I talk about multiple alleles, the most important thing is to remember the examples for it. And ABO blood group, you can expect problem based questions from this ABO blood groups, and this would be the first thing. And secondly, just concentrate on complementation test, recombination test. If you are not able to get in clear about what are these tests, at least know to interpret the results. Based on the result, just know like what conclusion you can derive from these results, and at least know about the significance, why these tests are actually used. So that would be sufficient. And lastly, the third point in this multiple allele chapter is the changing concept of a gene from classical concept, classical era to modern era. So how the concept of gene is changed, which is beautifully explained by Benzer. So if you are not going in detail about the experiment, at least know about the terms that was given by the Benzer and their meanings, like it is recon, cistron and muton and their meanings. So just try to know about them. So that would be enough for genes from the multiple alleles. Next we have sex determination. So these sex determination mechanisms are different for different organisms. So just make a note of all the different mechanisms that are involved, and like there are X and XY and XX, there are ZW and ZZ. So which are the organisms? Just know about the examples for all of them. We have haploid system and also we have diploid system that works in different organisms. Just keep a note of it. This would be the first thing to be done. And secondly, it's about sex index. So sex index is mainly based on genic balance theory given by Bridges. There is a table which tells about the sex index and associated sex, whether it is intersex, female, male, metamale or super female or super male. So there are 5 different columns or rows in this table. Just know about the index and associated sex with it. So try to remember these things. This would be very very important for you in the sex index under sex determination chapter. And also third, we have dosage compensation. So under dosage compensation, we have mainly 3 important mechanisms, that is inactivation, hyperactivation and hypoactivation. Know about the examples for all these mechanisms, in which organisms these mechanisms works. So if you are knowing this, that would be enough. After sex determination, it's about sex linkage. So it all starts with the experiment of Th. Morgan, which was in white eye gene for Drosophila, which is sex linked gene. So in this chapter, know about the non-disjunction concept and also about attached X chromosomes. So just read it in a very basic and superficial way. That would be enough from sex linkage. Next we have one of the most one more important chapter, that is mutation, where you can expect the questions. Just there are again 2 types of mutations that you can take. One is natural, another one is like induced mutations. So just know about how these natural mutations are occurring, and when we talk of the induced mutations, different chemicals which are used for inducing the mutations, and the classification, go through the classification once. After that, go through the mode of action, and lastly about the molecular mechanism or molecular basis of mutation. What changes does this mechanism brings in the at molecular level or at a gene or a chromosomal level? Just know about these things. Okay. And another important thing is to know about the transition and transversion. Why this transition and transversion is occurring, and most importantly, what does they mean? What is the difference between transition and what is one difference between transition and transversion? have a clear-cut understanding about these two terms, because you can directly expect the questions from these two terms. And lastly, we have cytoplasmic inheritance in genetics, that is in Mendelian genetics. So what you should cover in cytoplasmic inheritance is have a clear-cut differentiation between maternal effect and maternal inheritance or cytoplasmic inheritance, and examples for these two. If you cannot go through all the different mechanisms or the examples that are given in the book, at least know about the examples. Okay, try to cover the examples and revise them, and also know about the scientist who actually gave this different like cytoplasmic inheritance in different crops or organisms. So if you are going through these things, that would cover the whole Mendelian genetics part. Next we can discuss about molecular genetics. See, I will just oversimplify the things in the molecular genetics, because you cannot like you don't usually, like they won't be asking much difficult questions or much in-depth conceptual questions in the molecular genetics for PG exam or JRF. So you have to know about genetic material first, its properties. Before that, the proving of the genetic material, the experiment that were involved, the organisms, and the scientist, associated scientist. So this is the first thing about the genetic material. Secondly, it's about how the genetic material is replicated, that is complete mechanism of replication. Have the clear cut understanding about the replication. So once the replication is covered, then go for transcription and then translation. So just know about the initiation, elongation and termination in both the phases, and what are the different organelles that are involved in the translation, and the enzymes for both transcription and translation, and different factors. Okay, so just make it simplified, just go through the examples, like go through the enzymes, factors, and also the different organelles, and at which part of the cell this transcription and translation is occurring. This will be more than sufficient for molecular genetics. If you have sufficient time, just go through the regulation ones. I believe like you cannot cover this regulation part in full just by reading the BD Singh if you are reading it for first time or second time. Just keep your understanding to the basic level. At least know that why this regulation is occurring, and which are the, if there are any different enzymes or proteins that are involved, or different mechanisms or the process like phosphorylation, methylation. So just know about them. Okay, how they will affect on, what type of effect they will have on a character or a trait. So that you should know. If you are covering this, that will cover the molecular genetics. And also we have extra topic called as transposable elements, which is again very important. Just know about the examples for all the different types of transposable elements and their classification. So based on the mechanisms, they are classified into different types. Know about the classification and also about the examples. Okay, so this is what I wanted to cover from the genetics. See, in 15 days you cannot expect to cover more than this if you are starting now, or if you are in the middle of your preparation. Only if you are covered everything and if you have your own notes, then go revise everything, but prioritize these things which I have said you now. Okay, in the breeding, so starting with the history. So again from the history you have to cover the achievements of the and the important scientists and their achievements. So from the historical part, so that would be enough. So second unit, it is about germplasm conservation, where it is in situ and ex situ. Know about the in situ conservation of the and the ex situ conservation and the examples for them, and also gene pool concept. Gene pool concept is also very important, like we have primary, secondary and tertiary based on the cross compatibility. Know about these three categories under gene pool. And thirdly, it is about the centers of origin. Centers of origin, which are started with the 8 and which has now been modified into 11. Know about the centers of origin. So that will be enough for the second unit. Okay. So after the second unit, the third one is plant introduction. From the plant introduction chapter, you have to cover different types like primary introduction, secondary introduction, and the achievements, and what were the achievements of the introducing the plants from outside. We have secondary introduction where the green revolution, it was responsible for the green revolution, and you have to know about the Swamidwar varieties of rice and wheat, and how these varieties actually contributed for the green revolution. That would be important from plant introduction. And also the drawbacks. What are the drawbacks? What were the drawbacks of the introduction? Like they are associated with the weeds, pests, and diseases. Just read about these things from plant introduction chapter. Okay, so next is modes of reproduction. So from the modes of reproduction, like it is sexual, asexual. So from asexual, know about the vegetative propagales that are used for the propagation, and also about the gametophyte development. This is, I have covered these things in the seed physiology. Just go through those videos once. Or floral biology. In case of floral biology and seed physiology, I have covered this gametophyte development, that is megaspore, microspore development, all these things. Just read it in the BDSing once, and also cover if possible, cover through the videos from the YouTube. Okay. Next we have self-incompatibility. So from the self-incompatibility chapter, gametophytic types like gametophytic and sporophytic self-incompatibility, like these are the types and examples for all these two different kinds. So make sure to remember them, and the difference between the gametophytic and sporophytic self-incompatibility. So these things are important. And next, this is the first thing to be remembered, and secondly, you have to remember the techniques for elimination or suppression of self-incompatibility. For example, we use bud pollination or end season pollination, and we have several other mechanisms. Just keep a track on it. Okay, so that would be enough for self-incompatibility. Next we have male sterility. You know that male sterility is commercially used in case of hybrid seed production. So understand about GMS, CMS, CGMS, and different lines like ABR lines or ABR system, and how it is used in HSP, that is hybrid seed production. And also we use chemically hybridizing agents to induce the male sterility. Know about their examples and different chemicals and their mode of action. Next we have quantitative genetics. So what are the important concepts or the topics that are to be covered from quantitative genetics? From this quantitative genetics, know about the multiple factor hypothesis, formula like 2n plus 1 to find out the total number of phenotypic classes for quantitative trait. Remember this particular formula, and also types of genetic variants and their formulas. So if you are covering these two things, that would be more than enough. Next we have biomedical genetics and population genetics. So one thing I have not said in the genetics part is about the population genetics. So RD-Weinberg equation is the most important for you, because you can expect at least one question if they are asking questions from problems. So you can directly expect the question from the equation, that is RD-Weinberg's equation. So make sure to revise them and solve some problems related to this RD-Weinberg's law, and also factors which are affecting this RD-Weinberg's law. So these things are important in population genetics. Coming back to biomedical genetics, just know about the important designs and the scientists who gave them. The important designs that are used in the breeding and the scientists who are associated with them, just know about them. That would be sufficient. Next we have breeding methods. Again I will simplify these things. So from the breeding methods, just differentiate them based on self and cross pollinated. Know about like what are the different breeding methods that are specific to self and which are the methods which are specific to cross, and also the important procedures. Just know about the procedures, like how they are used, and important techniques in every method. Make a list of the method and know about the important techniques and important procedures. For example, like backcross methods is generally used to transfer
of oligogenes, right? So this is the particular thing that you should remember. And next we have pedigree method, where this is used for transgressive segregates. So there are certain things that you have to specifically remember from breeding methods. Just make sure to do it. Otherwise, just differentiate them based on self and cross, and just try to remember them. And one thing in breeding methods is to know about the scientist who has given the method. Know about the scientist. That would be very much important for you. Next, about heterosis breeding. From the heterosis breeding, just know about the formulas for heterobeltosis, like standard heterosis, mid-parent heterosis, or average heterosis. So all these things, just know the formula and revise them. And most importantly, it is about inbreeding depression. So know about the formula for inbreeding depression. And also you can expect the question from prediction of performance of double crossover, giving the single crossover from the parents. So know about the formulas for these things. Formulas for heterosis breeding. Okay, so this is all about heterosis. Next we can talk about breeding for biotic and abiotic stress. So from this particular chapter, know about the important donors, like they are present in the table form. Just go through the tables and know about the examples and genes from where they are isolated. For example, like freezing resistance gene is isolated from the fish. So like this, you have the certain examples, just know about them. And also we have certain important terms called as avoidance, tolerance, and resistance, and antibiosis and anticyanosis in case of insect resistance. Just try to understand the meaning of all these terms. And important genes for resistance, overall resistance or tolerance, you can this would be the first thing to be done. And secondly, just cover the gene for gene hypothesis in this particular chapter. So if you are covering these two things, that would be sufficient. So this would be the first thing from biotechnology. And secondly, cover the markers. The markers are also equally important, but don't dive deep into it. Just know about the dominant and recessive markers and the principle behind them, like a principle behind each of the marker if possible. Otherwise, stick to the dominant and recessive markers and their full form of different markers. Okay, next we have molecular techniques in them. Under this molecular techniques, cover the vectors, recombinant DNA technology, and like we have agrobacterium mediated transfer, which is the indirected type of transfer, and we also have direct type of transfer like electrophoresis, gene gun. So know about all these mechanisms or the process in case of recombinant DNA technology, and also read about the vectors and plasmids. So these would be sufficient for breeding. So just make sure to revise the things from the appendix once you cover all these things. See, if you are able to cover all these things, it is fine. If not, at least cover the appendix, or along with these things, try to cover the appendix. I would suggest you to cover these things first and go to the appendix. There will be a simplified form where you have to just remember them. So memory based things will be there in the appendix of BDC, both in breeding and also in genetics. Try to cover these things from appendix part. Don't forget to do it. Okay, once you finish all this content or digest all this content, make sure to revise the things from the appendix where it is clubbed and given in a single place. Okay, so now we shall continue with plant pathology. So plant pathology, these subjects we have dealt in detail in our master course from AgriAddict and also in the subject specific course. But since I was talking about 15-day strategy, you cannot follow all this content now. So for convenience, I will divide this plant pathology into 4 units. So to make it more simple and clear, starting with history of pathology and pathogens, and introduction to pathogens. So this will be the first unit. Second unit, it's IDM, integrated disease management. Third one, diseases of
agriculture crops and diseases of horticulture crops. So these are the 4 units. So I will just discuss each of them one by one, okay, in detail. So starting with history of pathology, just know about the important, like just know about all the scientists, like timeline of events that have been taken place along with the years, scientists and their major contributions. So if you just look into all these things, that will be covering the history of pathology part, and also it also covers the pathogens that were discovered from order, like order of their discovery. You can cover all these things from this historical part. Next we have introduction to pathogens. This is again one of the important unit to be covered, since you can expect some questions from this particular unit. So introduction to pathogens, they are mainly fungi, bacteria, virus, and we also have mycoplasma, phytoplasma, and we have spirilloplasma. So these are minor, okay. So these three, just know about the diseases associated with these pathogens in different plants, know about the examples, so that would be more than sufficient. So next, starting with fungi, it's all about the classification which you should concentrate on, like the order of to which this particular genus belongs to, which is causing the diseases, and the species, directly the disease and its scientific name, and behind that the order, to which order it belongs or to which phylum it belongs, and also know about the family. So these are the important things from the classification of the fungi, and also concentrate on the different types of sexual and asexual spores that are produced by different genus of pathogens. That would be the first thing, and secondly concentrate on fruiting bodies. So if you just cover all these things, classification of fungi is finished. Next, talking about the bacteria, know about the gram positive and gram negative bacteria, the classification and the examples. We have a few pathogens which are from the gram positive bacteria, know about these specific examples. So these things would be sufficient for bacteria, right? And uh, for virus, it's all about positive sense, negative sense in case of RNA, whether it is single stranded RNA, double stranded RNA, or it is DNA, whether the genetic material is RNA or DNA. Know about the difference and the examples for both the cases. For all these cases, know the examples, and also know about the different viruses under a single stranded and double stranded RNA and DNA. So if you cover these things, then it's fine. This was all about the fungi, bacteria, and the virus, and also other minor pathogens. So this would be finishing your first unit, that is introduction to pathogens or history of pathogens and pathology, and introduction to pathogens. Second, talking about IDM, know about the chemicals and different management practices that are specific to the crops, if any. Know about the have a general understanding about the fungicide, insecticides. Insecticides you can include here only with along with the fungicides, and the complete list of chemicals that are actually used to control the diseases, and also learn about different cultural methods. If you get specific cultural methods which are again specific to the crops, know about these things. So that would cover the idea. Next is diseases of agriculture, and I will just club these two things, disease of agriculture and horticulture crops. So what you should concentrate on these things, first thing is scientific name of the diseases of to whichever crop it belongs, and next it is etiology, that is symptoms. Just concentrate on the symptoms, and also few of the diseases will have a designated symptoms which will be highlighted in the bold letters. So just know about these symptoms, and the epidemiology, know about their spread, like primary and secondary source of inoculum, and lastly about the management practices. Again, these management practices can be very specific to the crop or diseases, know about them, and also know about the generalized management practices along with the different cultural practices if they are available. So these things will cover the complete pathology. If you are just focusing on these things, you can easily score like maximum marks in pathology, from pathology. Okay, so next we will discuss about microbiology. So for microbiology, I have made a detailed video on what are the important topics that that are needed to be covered. So the link will be provided in the description box. Just go and check the video for the details. It is actually 10 to 12 minutes of the video. It will definitely help you. So check out the video for microbiology. Next, it is for seed science and technology. So for seed science, I hope like many of you already knowing about the free lecturer series that I have uploaded in the YouTube. Again, the playlist link will be given in the description box. Go and check the video, which has only 16 videos where I have covered in detail of all the things, all the content that are very much required for JRF. So if you just go through these videos, if at least if you are covering 5 to 6 videos per day, we'll be finishing it by 3 days. So that would be the best thing that I can suggest you, other than just telling you again what are the important things, because I have covered in detail about the important topics or important content to be covered and explained everything in detail. So just don't forget to check out the video. Okay, so this is for microbiology and C-Tech. Anyhow, it is very simple and convenient to understand. And talking about other subjects other than these four major subjects, see what are the other things that you have to concentrate on. So other things is few topics from crop physiology, like photosynthesis, respiration, and nitrate assimilation. Again, this is again covered in microbiology also. Just go through it once again. And also plant hormones. Anyhow, this is also covered in C-Tech, in seed physiology. Just if you get extra information from anywhere else, just try to cover it. Like importance should be given to the functions and the roles and the site of their synthesis. So these will be important for plant hormones or plant growth regulators. So this is for crop physiology. So from biochemistry, just take care of biomolecules, have a clear understanding of the structure of these biomolecules, their functions. So that would be more than sufficient if you're going for biochemistry. That is specific. Keep your stick to biomolecules. It's about carbohydrates, lipids, proteins, and vitamins. Just have some elemental knowledge about all these things. So this will serve the purpose. And other things like general agriculture, you can just blindly follow the Nemra Sunda. So you just revise, at least give 1 hour, maximum 1 hour for now, and just try to revise the content that you have already studied. If you have not done it, just give 1 hour, 1 hour of your daily time, and try to finish as much of syllabus, as much as syllabus that is required. So under general agriculture, focus mainly on agronomy, soil science, entomology, and economics and extension. Okay, so you might expect the questions from these particular subjects. So mainly concentrate on these subjects under general agriculture. So if you are doing it, so it's done. So for the 15 days, if you just follow this video, you will be able to cover genetics in 2 to 3 days, plant breeding in again 2 to 3 days. It will take you the 6 days. I think only 1 or 2 days is required for microbiology, and also you can cover the videos of C-Tech in maximum of 2 to 3 days. So if you're doing so, you will get hardly remaining with another 5 days. So again, for the next 5 days, you can revise the content and keep the discussion with your keep the discussion with your friends, so that you'll be remembering the things. Whatever you have forgotten, you can again come back and revise. So and along with these things, you can also attend the mock test from whatever platform that is already available. So we are also providing the mock test. So you will get mock test for just 99 rupees. That is 18 full sets of mock test, and this is available in AgriAddict website, that is www.agriaddict.in. If you visit agriaddict.in, so you will get 18 full length papers plus one extra bonus paper for plant sciences just for 99 rupees. You can just go and opt it. Otherwise, if you're looking for chapter wise mock test, so you can download our app, which is again AgriAddict, where we are providing the complete mock test bundle just for 200 rupees for now. So either you can choose this or from the website, or you can choose both if you are already prepared for everything. So these mock papers are prepared for by JRF and SRF toppers from IARI. So they are like very standard. So we have seen it. So you can just take it, and it will definitely help you in boosting your confidence and also help in revising the things. So if you have any doubts or if you need any clarifications on anything, you can join our WhatsApp group. We have a dedicated WhatsApp group for plant science and also for other 7 major subjects. The links will be provided in the description box. You can visit the link and also join the WhatsApp group, and you can directly contact me through the number that is available in the description box. Okay, thank you so much.